Distinguished colleagues and students, it's an honor to present our team's latest research, published in iMeta, demonstrating how low-acylated rough-type lipopolysaccharide from Akkermansia muciniphila combats diet-induced obesity by activating the TLR4 IL-23 IL-22 immune axis. Let me introduce our key player, Akkermansia muciniphila, or AKK, a mucus-colonizing gut bacterium. In healthy individuals, it constitutes approximately 3% of the gut microbiota. But its abundance plummets in obesity, diabetes, and cardiovascular disease. Because restoring AKK levels improves metabolic health and strengthens gut integrity. In fact, these benefits have led to commercially available AKK-based products in Denmark. Even pasteurized AKK effectively treats metabolic disorders. This suggests its structural components drive these benefits. Since pasteurization preserves the cell wall and AKK is gram-negative, we focused on lipopolysaccharides LPS. But not all LPS is created equal. Pathogens like E. coli have LPS with hypo-acylated lipid and 2-phosphate groups. In contrast, probiotic LPS, for example, from bacteroids often features pentaacylation and 1-phosphate. This low acylation, low phosphorylation pattern correlates with beneficial metabolic effects. Akkermansia muciniphila lacks genes for secondary fatty acid synthesis in its LPS pathway, predicting a low acylated LPS. We hypothesized this unique LPS structure underlies AKK's therapeutic benefits. Our approach: 1. Isolated and characterized LPS from the China strain A. Muciniphila HW07. 2. Tested its metabolic effects in vivo. Akkermansia muciniphila LPS has lipid that is predominantly tetraacylated, with both mono and bis-phosphorylated variants. This confirms a unique low-acylated lipid structure. Functional results in obese mice, administered a LPS 0.2 mg per kilogram, IP to diet-induced obese DIO mice. ALPS treatment, improved metabolism and liver health. Regulated glucose and inflammation, strengthened the gut barrier. ALPS treatment upregulated the expression of IL-22 mRNA in intestinal epithelial cells and induced the expression of antimicrobial peptide genes. The IL-22 pathway was activated. Then we verified the core mediating role of IL-22 through an IL-22 antibody blocking experiment. ALPS treatment significantly elevated IL-23 levels in mice, establishing the IL-23-driven pathway for IL-22 secretion by ILC3S. Using TLR4 antagonists, TLR4 reporter assays, and TLR4 knockout mice, we confirmed specific activation TLR4 pathway, identifying TLR4 as the core receptor for ALPS. Thus, ALPS triggers IL-23 production in myeloid cells via TLR4, promoting IL-22 secretion from ILC3S. This signaling cascade TLR4 IL-23 IL-22 underlies the key metabolic protective mechanism of ALPS. ALPS reshaped gut communities, enriching beneficial bacteria linked to metabolic health, antibiotic depletion, and fecal microbiota transplantation. FMT proved ALPS requires the microbiota to improve metabolism and barrier function. In summary, our study elucidates the chemical structure of LPS from the Chinese Akkermansia muciniphila strain HW07 and defines its mechanism for combating obesity via the TLR4 IL-23 IL-22 axis. Given its favorable safety profile and immunomodulatory properties, this ALPS emerges as a promising novel therapeutic candidate for metabolic disorders such as obesity and diabetes.